Hello everyone and welcome. I'm Sean Sanders, Senior Editor for Custom Publishing at Science, and it's my pleasure to welcome you to today's Science AAAS webinar, Time-Resolved RNA Profiling for Cancer Research and Beyond. Conventional RNA sequencing, while allowing comprehensive transcriptome analyses at steady state, is limited for probing transcriptional responses to cell perturbations by the vast diversity of half-lives of the mRNAs and proteins being analyzed. At early time points, detectable changes in mRNA abundance are inevitably biased towards short-lived transcripts, while at later points, analyses do not allow for distinguishing direct from secondary effects. A simple scalable method for enabling direct detection of 4-thiouridine-labeled transcripts within the total RNA pool has been developed and can be combined with standard RNA sequencing methods for investigating dynamic changes in gene expression. This technique has been named thiol-linked alkylation for the metabolic sequencing of RNA, or Slam-seq. A key application of Slam-seq is to directly quantify specific or global changes in mRNA output following pharmacological or chemical genetics gene perturbation, and thereby define primary transcriptional functions of genes and drugs. I'm pleased to have the developers of this technique, Slam-seq, on the line with us today, Dr. Stefan Ramirez from the Institute of Molecular Biotechnology and Dr. Johannes Struve from the Research Institute of Molecular Pathology, both in Vienna, Austria. Many thanks to both of you for joining us. Before we get started, I have important information for our online viewers. At the top right of your screen, you'll find photographs of today's speakers and a view bio link that you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentations, we will have a short Q&A session during which we will address some of the questions submitted by our live online viewers. So if you're joining us live, start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Lexogen for sponsoring today's webinar. Our speakers today are going to present in quick succession, so I'm going to introduce both of them to you before they start their presentations. Dr. Stefan Ramirez joined the Institute of Molecular Biotechnology of the Austrian Academy of Sciences as a group leader in 2012. There he studies the molecular mechanisms governing small RNA silencing pathways in fruit flies and mammals, and the regulation of microRNA biogenesis and function by post-transcriptational epitranscriptomic modifications. The Ramirez lab is also developing novel epitranscriptomic technologies that can probe post-transcriptational gene regulatory networks at a transcriptome-wide level. Dr. Johannes Struve is a group leader at the Research Institute of Molecular Pathology and an adjunct professor at the Medical University of Vienna. He develops and applies innovative functional genetic models and screening technologies to identify and mechanistically probe dependencies and candidate therapeutic targets in leukemia and other cancers. A particular focus is the study of bromodomain-containing proteins and other chromatin-associated dependencies in leukemia. We're going to start off with Dr. Ramirez. Thank you so much for being on the line with us today.
Thank you very much, Sean, for the introduction. So today, we would like to introduce you to a novel gene expression profiling approach that adds a novel dimension to the analysis of standard high-throughput RNA sequencing approaches. And that's the dimension of time. And in doing so, time-resolved RNA sequencing provides rapid insights into the molecular basis of how gene expression profiles are established in order to study the regulation of gene expression at the transcriptional and the post-transcriptional level. But first, let me remind you that for cells to fulfill their biological roles in the context of tissues and organs of a multicellular organism, as displayed here, they need to mount specific gene expression profiles, and failure to do so can cause diseases, including cancer. So gene expression profiling by high-throughput sequencing of RNA has really revolutionized the way we can associate distinct gene expression profiles to specific cellular states, therefore providing mechanistic entry points to understanding the contribution of gene products to cellular function in the context of various physiological and also pathophysiological processes. However, currently available RNA-Seq approaches really only provide a snapshot of the relative abundance of any given transcript at a given time point inside the cell. So what they do not provide is the information on how these gene expression profiles are actually established. And that means the rates at which each of these gene products are produced and the rates at which they are degraded. But it's actually this information that provides us with insights into how each transcript is regulated, both at the transcriptional level, which determines the amount of RNA that is produced in a given time frame, as determined by a combinatorial action of promoters, enhancers, associated transcription factors, or chromatin landscape, to name but a few. And on the other side, at the post-transcriptional level, which determines the amount of RNA that is decayed over time, as determined by RNA binding proteins, RNA modifications, small RNAs, RNA structure, codon usage, and many more. Now, to provide rapid and robust insights into intracellular kinetics of RNA transcription and turnover, we have developed recently a metabolic RNA sequencing approach, which we refer to as SLAMseq. Now, this is a project that was led by a postdoc in the lab called Veronika Herzog, with support of a PhD student, Brian Reichholf, two very talented members of my lab. So SLAMseq stands for thiol-linked alkylation for the metabolic sequencing of RNA, and it is based on a well-established metabolic RNA labeling approach that employs the nucleotide analog 4-thiouridine. Now, 4-thiouridine is readily taken up by cultured cells through nucleoside equilibrate transporters and then incorporated into newly transcribed RNA. Now, under well-controlled experimental conditions, such metabolic RNA labeling in cells by 4-thiouridine does not affect cellular viability or gene expression because the nucleotide analog is biologically inert and to a large extent indistinguishable from unmodified uridine for the cell. Now, in a pulse or pulse chase metabolic RNA labeling setup, total RNA in the SLAMseq protocol is then prepared at given time points, and instead of previously described biochemical fractionation approaches that are typically time and labor intensive, require enormous starting material, and frequently suffer from poor or variable signal-to-noise performance, SLAMseq employs a simple thiol-specific chemical modification strategy that is based on the primary thiol reactive compound uracetamide. Now, in this protocol, uracetamide reacts with a thiol group of a 4-thiouridine and attaches a carboxyamido methyl group to 4-thiouridine by nucleophilic substitution, a reaction that results in close to complete alkylation within as little as 50 minutes reaction time, under conditions that, of course, do not affect RNA integrity and stability. Now, as a consequence, alkylation of 4-thiouridine changes the base pairing properties of 4-thiouridine, which now base pairs with guanine instead of adenine. Therefore, during reverse transcription in any of the standard cDNA library preparation protocols, results in a specific T to C conversion at the sites of 4-thiouridine incorporation. Now, the efficiency of this approach is highlighted in this example that you can see here, where we sequence a synthetic RNA that contains a 4-thiouridine residue at a single site. Now, SLAMseq enables to uncover this modified 4-thiouridine residue at single nucleotide resolution at an efficiency of greater than 94% by inducing a specific T to C conversion. Now, to effectively quantify metabolically labeled RNA in SLAMseq datasets, two talented bioinformaticians named Philipp Reschneider from the Max F. Perutz laboratories in Vienna and Tobias Neumann from the IMP in Vienna have developed a bioinformatics pipeline that we refer to as digital unmasking of nucleotide conversions in chimeras, or DUNC. And while I don't want to go into detail here, I just want to mention that this approach includes multiple elements that solve most of the challenges associated with the analysis of nucleotide conversion containing high-throughput sequencing datasets, including conversion-aware mapping to reference genomes, a multi-mapper reconciliation strategy to account for the reduced complexity of nucleotide sequence in reads, a SNP calling and masking approach, and finally, this in a combined manner enables to confidently deconvolute T2C conversion containing reads. Now, this is a pipeline that is also available via GitHub as indicated here. Now, while Slam-seq is compatible with any standard RNA sequencing approach and library preparation protocol that involves a reverse transcription step, the following brief examples that I would like to show you were generated using the mRNA 3' sequencing method called quant-seq, which is a rapid, robust, and cost-effective high-throughput gene expression profiling approach. Now, there are some key advantages of this mRNA 3' sequencing approach over whole transcriptome sequencing that I just want to highlight briefly. On the one side, it uses total RNA as input, therefore does not require any prior poly-A enrichment or RNA depletion. Library preparation protocols follow a robust and rapid workflow, which enables to generate sequencing-ready libraries in less than 5 hours, and the protocol can easily be scaled through automated library preparation protocols. quant-seq also enables a simple gene counting because any normalization to gene length is obsolete because only a short fragment is produced per transcript from the 3' end, which also enables to which also provides a lower overall library complexity, which enables to sequence libraries at lower depth and therefore enable cost-effective multiplexing. Furthermore, the bioinformatics analysis is facilitated because, for example, exon-exon junction detection is not required. And finally, high coverage of mRNA 3' tags facilitates the calling of T2C conversions because amplicons derived from the 3' UTR of mRNAs are inherently AU-rich. Now, there are also some limitations, of course. For example, the analysis is, of course, restricted to polyadenylated RNA species. That means RNA pol 2 transcripts, and it does not provide any information on splice isoform or transcript start sites. Now, here you can see an example of data generated via the quant-seq protocol derived from mouse embryonic stem cell total RNA. Now, what you see is a meta-analysis of quant-seq libraries across RNA polymerase II transcripts, which show enrichment of fragments within a window of around 250 nucleotides upstream of the mRNA 3' ends. And when we combine this sequencing approach with Slam-seq, you can see that after 24-hour labeling of cultured cells, in this case mouse embryonic stem cells as indicated, with phosphatidylcholine, T2C conversions shown in red are significantly elevated compared to background sequencing errors, which typically range below 0.1% for the Illumina sequencing platform. And T2C conversions distribute evenly within the region of cover, within the region that is covered by sequencing. Now, Slam-seq experiments can actually come in two flavors. First of all, an anabolic RNA labeling approach, which is basically a pulse labeling experiment, which provides insights into RNA synthesis kinetics by fading out the differential effect of transcript stabilities. And it can also be employed to determine RNA processing kinetics. The second type of experiment is a catabolic RNA labeling approach, where transcripts are labeled to saturation, followed by a chase with unlabeled uridine to follow the degrees of labeled transcript over time, which enables to study RNA decay kinetics. Now, while we focus on the analysis of transcriptional regulation in cancer cells in the second part of this seminar, which will be covered by my colleague, Dr. Johannes Zuber, I would like to briefly illustrate how to measure transcript stabilities to study key concepts in post-transcriptional gene regulation using Slam-seq. Now, to this end, we subjected mouse embryonic stem cells to 4-thiouridine metabolic RNA labeling for 12 hours, followed by washout and chase using non-thiol containing uridine, and we prepared total RNA at various time points along this chase. Then total RNA was subjected to alkylation using the SLAMseq protocol and mRNA 3' sequencing using quant-seq. So first we focus on candidate genes. Here you can see 4 candidate genes to show some primary data. And here you can see a genome browser coverage tracks for these 4 genes with a zoom into the 3' end across the whole time course of the chase experiment. Now, when inspecting all reads, that means metabolically labeled and unlabeled reads together, shown in black here, we observe that steady-state gene expressions stay constant across the whole time course which is what one would expect if working under unperturbed conditions, as we did. In contrast, T2C conversion containing reads, shown in red here, decreased over time in a transcript-specific manner, which is independent of the steady-state abundance of each of the transcripts. After calculating the background subtracted U content and coverage normalized TC conversion rate for each transcript at every time point relative to chase onset, conversion rates fit well to single exponential decay kinetics, enabling the robust determination of polyadenylated transcript half-lives as indicated here. As expected, RNA stabilities differed by more than one order of magnitude among individual transcripts. For example, a short half-life was observed for the primary microRNA transcript of the miR290 to 95 cluster with a half-life of less than 1 hour, most certainly because it is subjected to a rapid co-transcriptional processing by the RNase-3 enzyme DROSHA in the nucleus. In contrast, particularly long half-lives were detected for housekeeping transcripts such as ribosomal protein encoding mRNAs such RPL10A, shown on the right side. Now, by fitting the data of more than 8,400 transcripts with a steady-state expression cutoff of more than 5 reads per million, which is an arbitrary cutoff and can easily be further reduced, to single exponential decay kinetics, we determined a median mRNA half-life of 3.9 hours, corresponding to a cell cycle normalized half-life of 4.2 hours, which falls within the range of previously determined mRNA stabilities in mammalian cells, ranging in median half-life from 3.4 hours to 10 hours, depending on cell type and an experimental approach. Now, these data also confirm a close relationship between transcript-specific mRNA half-life and its physiological function. Because when we ranked the transcripts according to their relative stability and performed gene ontology enrichment analysis for the 680 most or least stable mRNAs, we found that transcripts encoding proteins with housekeeping functions, such as protein synthesis or respiration, tend to decay at low rates, perhaps reflecting the evolutionary adaptation to energy constraints. In contrast, transcripts with a regulatory role, such as transcription factors or cell cycle genes, tend to decay much faster, most certainly providing control because this provides control of the persistence of genetic information and therefore facilitates adaptation to environmental changes. Now, therefore, Slam-seq measurements recapitulate key concepts in post-transcriptional gene expression control and transcript stabilities. Now, because we use an mRNA 3' sequencing strategy, we can even assign transcript half-life to each of the 3' isoforms of a given gene locus, as illustrated here for this gene called ARFRP1, where we can see a more than 5-fold difference in mRNA 3' isoform stability. And it illustrates how we can start to understand the differential regulation of transcript isoforms. Now, with this, I would like to briefly summarize the first part of this webinar and give a short outlook on the scope of metabolic RNA sequencing beyond the measurement of gene expression dynamics. Now, I have introduced you to a metabolic RNA sequencing by Slam-seq, which is an orthogonal chemistry-based sequencing strategy that quantitatively uncovers thiol-uridine incorporations in RNA species at single nucleotide resolution. When coupled to well-established thiol-uridine metabolic labeling protocols and standard RNA sequencing approaches, such as mRNA 3' sequencing, Slam-seq enables rapid access to gene expression dynamics in living cells at unprecedented accessibility, efficiency, and scalability. I've briefly exemplified this by showcasing a pulse chase labeling scenario in mouse embryonic stem cells, which enables the determination of gene-specific and global transcript stabilities. And while I didn't have time to show you today, this approach can be readily combined with genetic perturbation settings to study the contribution of individual gene regulatory pathways to overall transcript decay. Now, in just a minute, Johannes Suber will introduce you to how Slam-seq can be employed to identify primary transcriptional targets of regulatory genes or pathways in order to, for example, study transcriptional regulators in cancer. More recently, we could also collaborate with the labs of Eric Miska at the Gurdon Institute in Cambridge and Oliver Rando from the University of Massachusetts Medical School in Worcester to combine previously described cell type-specific metabolic RNA labeling approaches in vivo in mice using the Cre-Lox-dependent expression of the Toxoplasma gondii enzyme UPRT and systemic application of thiol-uracil, which enables to determine tissue and cell type-specific gene expression in vivo in mammals. If you want to know more about this, I have the reference indicated. And finally, a similar approach can also be applied to determine the transport of RNA species between cells and tissues in vivo, as exemplified by Oliver Rando's lab in the case of RNA transport from the epididymal epithelium to the developing sperm in vivo in mice. Now, with this outlook, I would like to thank you very much for the attention, and I would like to hand over to my colleague Johannes Zuber for the second part of the webinar, where he will focus on the identification of primary transcriptional targets of regulatory genes and pathway in cancer using Slam-seq.
Yeah, thank you, Stefan, for the introduction, and welcome also from my side. So our lab is particularly interested in the study of transcription factors and chromatin regulators in cancer, not only because these genes are very frequently mutated in this disease, but also through various approaches we and others have identified very promising dependencies and potential therapeutic targets in this gene space. For some of these regulators, actually small molecule inhibitors have already been developed and have advanced at certain stages on the to clinical investigation. So one key information we're really missing to understand the function of these genes and advance these molecules towards clinical use is really the set of genes these factors are regulating directly in the genome. And that's not an easy question to answer, and today I want to showcase you a few examples how Slam-seq can actually advance this knowledge very rapidly. So for determining direct genes, established techniques cause various challenges. Chip sequencing is not predicting regulatory relationships in the genome, and conventional mRNA sequencing really doesn't allow us to distinguish primary from secondary effects after perturbing a gene. And these challenges are very well illustrated by one factor we've been studying over the past years, and this is BRD4, histone acetyl reader, which very broadly binds actively transcribed genes, both at the promoter and enhancer level, and which can be inhibited by beta bromodomain inhibitors, which are currently under clinical investigation. Now, in contrast to the broad binding of BRD4, old mRNA sequencing approaches we've performed previously always showed rather selective transcriptional responses to beta bromodomain inhibition at various time points. Now, if we do these experiments and think about actually what we're looking at here, we came to realize that there is simply no perfect time point to determine direct transcriptional responses to beta bromodomain inhibition in a definitive way, because if we sample at very early time points, inevitably we will be biased by short half-lives. And if we look back actually at our previous RNA sequencing data, we see that these short time points were heavily biased for short-lived transcripts. Now, at later time points, we lose these biases, but again, inevitably we'll sample a lot of secondary effects, and this is particularly severe in case of BRD4, where we know one very prominent target of beta bromodomain inhibitors is the transcription factor MYC. And as Stefan has already pointed out, as many other transcription factors, MYC is very highly turned over. We get protein suppression already after a couple of hours, and anything we sample after this time point could be just a secondary effect of suppressing MYC. So very clearly, looking at the total abundance of mRNAs in the cytoplasm is insufficient to actually distinguish primary from secondary targets. So we need to look at mRNA production, at mRNA output following drug treatment, rather than at changes at the total mRNA level. And this can be done through 4SU labeling, as Stefan already has introduced, but the method he just described really, we believe, revolutionizes the scale at which we can do these experiments, because here in Slam-seq, we're using a very simple chemical trick to, very simply put, make 4SU visible in next generation sequencing of total RNA. So as soon as we learned about this chemical trick from Stefan, we got very excited about developing this as a tool for probing transcriptional responses to cell perturbations. And for this, three very talented people in the lab developed protocols that are specifically suited for these short label time points, where we usually label only 1 to 2 hours to exclude the possibility of secondary transcriptional effects. We end up with a very simple and very scalable protocol that can be done for many samples in just a single day. And I want to show you some typical results you get after bioinformatic analysis using the Slam-seq analysis pipeline. So for each gene in the genome, you will get, as with usual mRNA sequencing, the number of total mRNAs. But besides that, you also get information about mRNAs that have been newly produced in this 1 hour of 4SU labeling. And as you can see here, for average genes, the abundance of these new mRNAs ranges somewhere between 1 and 10%. Now, very convincingly, already in the first Slam-seq profiles we did, we could identify that one of the most highly turned over transcripts actually turns out to be MYC, which we knew as a very high turnover transcript before. We find that after 1 hour of labeling, 75% of MYC transcripts actually contain T2C conversions, flagging them as newly produced within the hour of labeling. And we can actually, from these numbers, roughly, but actually quite accurately estimate transcript half-lives already. And if we look at this globally, we see that only a small fraction of genes has such high turnover kinetics. Unfortunately, these are very enriched for transcription factors, really illustrating the problem we are having if we are sampling too early with total mRNA sequencing, resulting in large biases, and at later time points, we would capture a lot of secondary effects from perturbing these genes early on. So as a next step, we turn to now drug treatment, where we perform basically 2 Slam-seq experiments in parallel. One we just treat with DMSO, and one with an inhibitor. After pre-treatment of about 15 to 30 minutes, we label again for 1 hour with 4SU, and now can directly measure the changes in mRNA output. We started out with treatment experiments using inhibitors of CDK9, which are predicted to globally stall transcriptional pause release and thereby trigger a global repression of transcription. So if we look at total mRNA reads after this 1 hour, we see that for most genes we cannot detect any major effects. Only for the short-lived MYC, again, we can already see a small decay of abundance of mRNAs. However, if you look at Slam-seq, we see that independent of mRNA half-lives, we see a complete shutdown of transcription, as we would expect from CDK9 inhibition, completely independent of mRNA half-lives. So as a next step, we turn to more selective inhibitors, which we can profile for their direct transcriptional effects. And as we're working in K562 leukemia cells, which actually harbor a BCR-ABL mutation, we had several pathway inhibitors that we could analyze comparatively. So for each of these inhibitors of signaling pathways in downstream of the BCR-ABL fusion protein, we could see very selective responses with great precision, and one can already derive some quite clear conclusions from these data. For example, that the oncogene MYC is entirely regulated and exclusively regulated through the RAF-MEK-ERK signaling pathway, not affected by the PI3 kinase signaling pathway. If you compare these profiles, they are very distinct from each other, telling us how selective Slam-seq profiles can be with selected pathway inhibitors. But if we now combine the 2 inhibitors inhibiting pathways, key pathways downstream of BCR-ABL, actually the effects of this perturbation very much approximates to the inhibition of the upstream kinase, which illustrates that Slam-seq can actually be used to understand the hierarchical wiring of signaling pathways or annotate inhibitors where they act in such hierarchies. So let me quickly summarize this technical part. Slam-seq, we believe, revolutionizes the annotation of direct transcriptional responses to cell perturbations. We like the fact that Slam-seq combined with quantiQ actually directly quantifies mRNA output, the product of transcription, instead of measuring intermediates. As I've shown you for CDK9, we can directly measure specific and global effects directly in total mRNA sequencing. We don't need spike-in controls for detecting global effects. But the really key advantage and advance of Slam-seq is its simplicity and scalability. It's compatible with regular cell numbers you use for RNA sequencing. It's a very simple one-day protocol, and you can handle many samples, really enabling multi-sample studies, which could be used, for instance, to perform dose escalation experiments, time course experiments, and also compare many different chemical entities side by side. For the technical implementation of Slam-seq, Steph Bannister from Lexogen will actually, after the Q&A session, take you a little bit more into details about solutions, how you can develop Slam-seq in your lab. So far I've told you about the use of Slam-seq for probing drug action of already established inhibitors, but unfortunately for many genes we want to understand functionally, we don't have such inhibitors in place. And that's even true for BRD4, because BET-bromodomain inhibitors are inhibiting several BET-bromodomain containing proteins. So for really probing the function of individual transcriptional regulators, we got very excited about combining Slam-seq with emerging chemical genetic protein degradation technologies, of which many exist, but we got particularly excited about the auxin-inducible degron system, which is a system which uses a degron tag from rice together with an F-box protein from rice that sees and recruits AI-detect proteins to the mammalian SCF complex for rapid degradation. And the key advantage we find for the auxin-degron system is really its speed. If you want to perturb a gene and perform Slam-seq experiment, you depend on techniques that allow very rapid gene perturbation, just like drugs. RNAi or CRISPR are, we believe, too slow of a perturbation. Auxin-degron technology really allows us to suppress proteins within minutes. And we've done this now for several nuclear proteins, here illustrated by BRD4. Within 15 minutes, we have already nearly complete degradation of this factor. And this degradation is very selective, specific even for proteins that reside in large complexes, such as BRD4. Now we can do an experiment where we degrade the factor and then measure directly the changes in transcriptional output, just as I've previously described for drugs. And in case of BRD4 degradation, we see a global repression, a very broad repression of transcription, very much in line with BRD4's proposed function as a key factor in pause release. We can align this also with other chromatin profiling assays we have done. We see that BRD4 indeed globally reduces elongation-associated serine 2 phosphorylated Pol2, as well as leads to a stalling of polymerase at the TSS stall site. So the next step was to understand now what is the difference between actual BRD4 degradation and BET-bromodomain inhibition. So for this, we now applied a relatively high dose of JQ1, and now for the first time, we could see with JQ1 indeed a very broad global repression of transcription at these high doses of JQ1, suggesting that BRD4's recruitment and function in the chromatin is largely BET-bromodomain dependent. And we can also see this by just looking at BRD4 chromatin binding following JQ1 treatment using Western blotting. So now 5 micromolar of JQ1 is an extraordinarily high dose. We wanted to understand what are relevant JQ1 targets at therapeutically relevant doses, which I think are very well determined in leukemia models. These are doses that have selective activity in certain hypersensitive cancer models. And here we see a much more selective response to JQ1 treatment. And actually, if we extended these analyses to now a variety of myeloid leukemia cell lines, we see that the scale of these responses is actually well preserved. And actually, these responses very well correlate between different leukemia cell lines and involve always quite a similar set of genes, of direct target genes. And that's really some point which we have previously missed because it was not detected by conventional mRNA sequencing. Now, one question that arose from these analysis was, is JQ1 therapeutic activity just simply the consequence of an incomplete interference with pause release machinery that also could be achieved by inhibiting other factors incompletely in this complex? And therefore, we compared now very quickly JQ1 responses, 2 responses to CDK9 inhibition, but now not as previously shown at high dose CDK9, which leads to a complete shutdown, but at CDK9 inhibitor doses that had similar cellular effects as the JQ1 dose that we believe has the therapeutic window. So now, if we now compare the transcriptional response between JQ1, low-dose JQ1 and low-dose CDK9 inhibition, we see that JQ1 leads to a very conserved response into various leukemia cell lines, while low-dose NBP2 CDK9 inhibition has a very distinct response. So inhibiting this complex at different stages triggers selective responses. We could also use now these two drugs to try to understand the mechanistic underpinnings of the strong synergy that has been described between these two agents. If we combine these two low-dose interferences with pulse release machinery, we again actually trigger a global effect, which we believe is the mechanistic explanation for their synergy and should raise some concerns about the toxicity of this combination. So to summarize this first part, I hope I could show you that we can use Slam-seq in combination with auxin degradation very rapidly to characterize the transcriptional functions of chromatin regulators as illustrated by BRD4, which we identify as a general cofactor in transcriptional pulse release. In contrast, therapeutic doses of BET inhibitors trigger very selective transcriptional responses that are very conserved, surprisingly conserved, between diverse leukemia contexts and not very strongly associated with the presence of superenhancers. So we also find, as one conclusion of this study, that MYC is indeed uniformly hypersensitive and a key target of BET inhibitors in leukemia. Its role as a direct BET inhibitor target in other cancers, I think, should be further investigated. But in leukemia, if one wants to understand the activity, the biologic activity of BET bromodomain inhibitors, one better understands what MYC is actually doing in this disease context. And there we're entering another very controversial field where MYC has been described as a transcription regulator in many different ways. It has been described as a very broad transcriptional amplifier of basically all actively transcribed genes, also as a selective transcription factor, both as a transcriptional activator and repressor. So we thought with the combination of auxin-inducible degradation and Slam-seq, we have a new way of asking this question and basically applied this technology in the same way I just described for BRD4, now for MYC, tagging endogenous MYC, degrading it very rapidly, which again worked with extremely high dynamics, and this has actually worked for many transcription factors we've meanwhile tagged and degraded in the lab, and then perform, following its degradation, a Slam-seq experiment, capturing changes in transcription output over the next hour. So if we do this Slam-seq experiment, just from looking at the primary Slam-seq plot, one can immediately appreciate that MYC, unlike CDK9 inhibition, unlike BRD4 degradation, and unlike high-dose BET inhibitor treatment, does not trigger global transcriptional responses. It appears to be a selective transcription factor, and the scale of response is actually comparable with other transcription factors we've meanwhile investigated. It also becomes clear that MYC, in contrast to many other factors we've meanwhile studied, is predominantly a transcriptional activator. We see very little evidence for direct repression of genes. And if you go into the ontology of the regulated genes, MYC very dominantly regulates genes involved in very basic synthesis processes, such as protein biosynthesis. MYC regulates about 50% of ribosomal components at the transcriptional level, as well as de novo purine synthesis, where all 6 enzymes constituting this pathway are actually very prominent direct MYC targets. We next ask how conserved these targets are in different cancer contexts. On the left side here, you see 2 independent AID-tagged clones in the leukemia cell line, K562. And on the right side, you see a correlation between one of these clones with a human colon cancer cell line. And you may appreciate that these transcriptional responses were highly conserved in these very unrelated cancers. And we've meanwhile extended this even to mouse models of leukemia, where we also can see that these effects are conserved across species as well. We can also take this further and now use the Slam-seq defined MYC response signature and ask for its correlation with MYC expression in, for instance, large collections of RNA sequencing data in cancer cell lines, where we see there's a rather good correlation already on the global scale, but we see many of these outliers that actually have a relatively low MYC expression, but still express that signature. And if you look closer into these outliers, virtually all of these outliers actually express N-MYC or L-MYC as paralogs of C-MYC, so suggesting that these paralogs can take over MYC's function in regulating these genes. We can take this even further into TCGA analysis of over 5,000 patient samples, where we can distinguish MYC high and MYC low expressing cancers in diverse cancer entities. And in every single cancer subtype we do these analyses, we see that the expression levels of MYC go very well along with the expression of this MYC target gene signature, making us believe that these are the relevant genes regulated by MYC across cancers. And this can be also seen in conventional GCA analysis, where again in every cancer this target gene signature is very strongly correlated with MYC expression. So to summarize this last part, MYC acts as a transcriptional activator of a confined set of target genes that are highly conserved between diverse cancer contexts. We see that MYC predominantly regulates genes involved in basic synthesis processes, including purine synthesis, ribosome biogenesis. MYC regulates several components of RNA polymerases, as well as other metabolic enzymes. And if one takes this together, this provides an explanation why after perturbing MYC, indeed, global mRNA and protein levels are altered in cells after several hours of MYC perturbation. But this is not due to a direct transcriptional function of MYC at these genes, but rather indirect effects from its biological functions. I hope I could also convince you that combining rapid protein degradation and Slam-seq provides a very simple and scalable approach to deeply investigate primary transcriptional functions of genes or pathways, and we are certainly excited to apply this to different factors and get a more detailed and definitive understanding about their primary transcriptional functions. With this, also in Stefan's name, I want to thank again our two labs. We acknowledged key people along the way, but this has really been a great team effort of both of the labs. We also want to thank our collaborators here at the Vienna Biocenter at Boehringer Ingelheim, and throughout the world. We want to thank our funding sources, and we're looking forward to your questions.
Thank you so much, Dr. Amiras and Dr. Zuber. It's now time to get to some of the questions submitted by our online viewers. A quick reminder to those watching us live that you can still submit your questions by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. And I'm sure our speakers would welcome your questions and inquiries. So let me put the first question to you, Dr. Amiras. What are some of the important technical considerations that I should consider when performing a Slam-seq experiment? And this is from a viewer who's just starting out and would like to try this out in their research.
Yes, so thank you very much for this question. I think, when one starts to consider a Slam-seq experiment, the most important point for sure will be to test the uptake of the nucleotide analog into the cells. And this is a point that needs to be experimentally addressed because depending on the cell type, uptake of phosphoryuridine might occur more readily or less readily. So we have tested so far a number of different cell types, mostly murine and human origin, but also of Drosophila origin, for example. And depending on the cell type, uptake can vary quite substantially. So what we typically recommend when starting a Slam-seq before starting a Slam-seq experiment is to do a titration series to measure the uptake and incorporation into RNA species. And this can be done in different ways. For one can do a small-scale Slam-seq experiment and sequence, for example, as a spike in a sequencing track. On the other side, one can use an HPLC type of analysis that we describe in the manuscript that has recently been published in Nature Methods, for example, where one can use total RNA digestion to a single nucleoside and then use an HPLC platform to test for the abundance of 4-thiouridine in metabolically labeled RNA. One thing that I should also mention, and this has also been reported in the literature, is that extremely high doses of 4-thiouridine can result in toxic effects. And this is a point that needs to be carefully addressed for any new cell type that where a Slam-seq or any metabolic labeling experiment is performed. So there we recommend, and there's also a titration kit setup available by Lexogen, that one titrates carefully the concentration of 4-thiouridine and then measures cytotoxic effects through standard cytotoxicity quantification assays that are also commercially available in order to test when toxic effects set in. Then it is extremely important to work at more than an order of magnitude in concentration below the EC50 value. This is our typical recommendation for setting up a metabolic labeling experiment and a Slam-seq experiment. Otherwise, the chemical conversion, I think, is very straightforward, technically not very demanding, and subsequently any standard RNA sequencing approach can be used that follows the standard descriptions from the commercial provider or the homemade approach if necessary.
Thank you so much. Dr. Zuber, a question for you. When using Slam-seq for monitoring drug responses, do you have a recommendation for the time points that should be considered for pretreatment and for 4-thiouridine labeling?
Yeah, thanks for the question. That's certainly an important point to consider. And I guess what I can tell you is just conceptual considerations and our so far experience with about 15 small molecule inhibitors. So the two times which are really important to consider is do I have to do any sort of pre-treatment? Which type of drugs actually are ideally suited? So any drug that would trigger, that you expect to trigger a direct transcriptional response is ideally suited for Slam-seq. There are other drugs which may not act through transcription or have delayed effects due to turnover, for instance, inhibitors of histone-methyltransferases. I would not predict an instant transcriptional response. Now, for drugs where I have an instant transcriptional response, we still prefer to pre-treat this drug for about 30 minutes, and that is simply due to the fact that transcription takes time. So for longer transcripts, we want to ensure that transcripts that have been initiated prior to drug treatment are basically run out before we actually start monitoring the changes in transcriptional output. The other important time point to consider is how long to label, and there are basically two parameters to consider. The lower end of labeling is determined again by the time of transcription. That again takes time, and also by the depth you want to sequence. So how many new mRNAs are produced, and the upper time point is determined by when I would conceive secondary effects due to mRNA turnover, protein turnover, and then secondary effects. And so what we like to do currently is labeling times between 1 hour and 2 hours, which have worked very robustly for us, and which we think is so 1 hour is probably the shortest you can do. 2 hours, we still feel comfortable that there will be very little secondary effects. So that's what we are currently using for drug profiling.
Thank you very much for that. So now a question I'm not sure who would best be able to answer this, so I'll put it to both of you, and maybe you can choose since you're on the same line. The question is Slam-seq sensitive enough to capture changes in co-transcriptational processing events?
Yes, so that's a very good question. I would say that in its current version, SLAMseq can measure the kinetics of any transcript that is detected at steady state. So if an RNA transcript is detected in a standard RNA sequencing approach, then we can also measure the incorporation of nucleotide, and therefore can detect it also by sequencing and measure the kinetics. What I have to mention in this case, of course, the way we have introduced it right now and it's currently published, we are applying a 3' sequencing approach. And this 3' mRNA sequencing approach primes at polyadenylated transcripts. So therefore, co-transcriptional processing events are not readily detected in RNA sequencing approach that measure through the quantify protocol, so mRNA 3' sequencing protocol, because only a short stretch of around 250 nucleotides from the 3' end of the transcript are actually sequenced. Now, having said this, SLAMseq is readily compatible with any type of standard RNA sequencing approach, for example, also ribose 0 RNA-Seq. And in these sequencing approaches, transcriptional byproducts such as introns, for example, are in parts readily detected, at least for abundantly expressed transcripts. And for these, of course, it is then also possible to measure decay kinetics, for example, of intronic stretches of transcripts. So I think that should answer the question.
Fantastic. Dr. Meres, are the methods applicable to other gene expression situations, such as a response to diet or external stimulus? And they follow that up by saying it appears that your focus has been in cancer regulation situations, but they foresee that this is possibly a bridge to more routine epigenetics applications.
Yes, absolutely, and we're certainly excited about such applications. One that is, for instance, immediately foreseeable is, for instance, responses to cytokines and other cell reactions that one can model. So, in principle, anything that you expect to directly impact, either directly or through signaling transcription, can be monitored. And the information you gain for it is that you have, over a very short period of time after, for instance, adding a cytokine to cells, or after restricting a certain nutrient, if you believe this is having an instant effect at the transcriptional level, that you get a precise measurement of changes in transcriptional output. We are certainly exploring such applications, and we're very excited about them.
Okay, Dr. Meres, let me give you one. How do you choose a reference gene for these experiments, since all RNA species are at some point degraded?
So I think this question goes to the point of if there are global changes in decay scenarios, for example, or transcript stabilities, whether SLAMseq can measure those. And the simple answer is yes, we can measure these without reference genes. And the reason for that is that label transcripts are measured in the context of total RNA. So that means that if in a standard catabolic labeling approach, so that means steady state labeling conditions for 12 hours, and then removal of the label and chase with cold, so unlabeled uridine. In these cases, we are basically following for each transcript the removal and the disappearance of the signal over time. So we do not need any reference genes because steady state gene expression doesn't change in this procedure. And therefore we can measure even for very long or very short-lived transcripts the stability irrespective of their either very long or very short half-life. And this is the key difference when it comes to previously established approaches such as pull-down approaches where one pulls only on labeled transcripts and measures the relative abundance of transcripts then over time. So I think that's the key difference in SLAMseq.
And I should add that also in the inverse scenario of anabolic measures, as long as your labeling time is restricted to 1 or 2 hours, as I said, you can use the total mRNA abundance as a reference because, or the median of the total mRNA abundance as a reference, because that's not predicted to change and actually does not change in such short labeling time points. So again, you have an ability to directly internally normalize and control and get actually, as we've shown for really one of the most brutal things you can do to transcription, inhibiting CDK9, you directly see a complete breakdown of transcription. So it's internally controlled as long for anabolic processes as long as your labeling time is short enough.
Fantastic. So we've got time for one more quick question. This is to both of you. I'm assuming you've tried this in a number of different cell types. Are there any cells that cannot be used to do Slam-seq experiments?
We have so far not found any cell type that are entirely non-amenable to 4-thiol nucleotide uptake and incorporation. That being said, for example, for very specific cell types, when it comes to catabolic labeling, S2 cells, for example, are problematic to wash out 4-thiouridine in a pulse chase scenario. So S2 cells, these are Drosophila. This is a Drosophila cell line. The reason, so this has been described in the literature previously. The reason for that is unclear, perhaps because there are vesicles that store 4-thiol inside cells, and therefore it's difficult to wash the nucleotide analog out. This is one possibility. It hasn't really been examined. Other than that, when it comes to all mammalian cell types that we've looked at so far, both anabolic and catabolic labeling approaches do not show any problems. And I think the same is true for Dr. Zuber's approaches.
Well, I should say, I mean, we've been very excited that a standard dose of 100 micromolar, which is actually not very toxic in a 24-hour assay to most cell types we've tested. It's somewhere in an IC50 in a, again, 24-hour, 48-hour assay. Has been working very well for us. We've just encountered one example where a lung cancer cell line has not efficiently incorporated with 100, but what I can strongly recommend there is to be not afraid to go up with 4-thiol, and this actually fixed the problem, especially for these 500 micromolar is the dose we would recommend in case 100 doesn't do the job. And 500 is indeed quite toxic, but again, after 48 hours, if you actually sample your cells after 1 or 2 hours of labeling, we don't believe this has any major effect on cell viability.
And I want to very quickly follow up on this because I did not mention this before. The toxic effect that we are talking about here is actually attributed very likely to an rRNA biogenesis defect. And this sets in at really high doses of metabolic labeling. So I think this does not globally interfere with RNA biology at various different levels, but it seems to be a very specific effect when it comes to non-coding RNA processing, such as ribosomal RNA, which makes really a large fraction of the overall total RNA content of a cell. And there seem to be defects that set in at very long labeling times, as Hannes already mentioned. So I think when one talks about catabolic labeling approaches, that's one point to consider. But in all the approaches that we've run so far in the lab, we can achieve conditions that are non-toxic to any given cell type, and that enables still to acquire signals in Slam-seq experiments that are sufficient to have to acquire robust metabolic sequencing results.
Thank you very much. And unfortunately, that's all the questions that we have time for today. But before we go, as Dr. Zuber mentioned, Dr. Stephanie Bannister from our sponsor Lexogen is going to briefly provide us with some further information about available products that are relevant to today's webinar.
Thank you, Sean. And I'd like to say Lexogen is very pleased to be sponsoring today's webinar on Slam-seq, and thank you all for joining for the final part of the presentation. So to quickly introduce myself, I'm Head of Applications and Customer Support at Lexogen, and today I'm just presenting a short overview of the Slam-seq kits that we offer, which are based on the Slam-seq technology that you've heard about in today's presentation. So our Lexogen Slam-seq kits allow you to perform Slam-seq RNA labeling experiments for any RNA sequencing applications. We offer two different types of kits, the first being the Slam-seq Explorer kit. In the Explorer kit, we have two modules available. The cell viability titration module is used to determine the 4-thiouridine dosage optimal for your chosen cell type and to optimize labeling durations. The S4U incorporation module provides additional solutions that allow you to prepare labeled RNA from cells and assess the level of 4-thiouridine incorporation using an HPLC assay. The Slam-seq kinetics kit modules contain reagents for 4-thiouridine labeling, RNA isolation, and RNA alkylation, and are designed for use with our single time point or time course labeling experiments. The anabolic kinetics kit module enables pulse labeling experiments, facilitating analysis of RNA synthesis kinetics, while the catabolic kinetics module contains additional unlabeled uridine, which allows you to perform extended labeling with 4-thiouridine, followed by a uridine chase, and this is in order to measure RNA degradation kinetics. So all of these kit modules are available in a 24 reaction size and based on a 24-well cell culture format. However, please contact us via info@lexogen.com if you're interested in other kit sizes or formats. So the Slam-seq kits that we offer enable you to label your cells and prepare alkylated total RNA for your Slam-seq experiments. As you've heard in both presentations today, RNA labeling and alkylation performed using Slam-seq is ideally combined with quant-seq 3' mRNA-Seq library prep. This can be used for a range of applications, including RNA kinetic studies and cellular perturbation assays, which you've heard about today. And you can easily combine any of our Lexogen Slam-seq kits for RNA labeling and alkylation with our quant-seq forward and reverse kits for library preparation. Using quant-seq for your Slam-seq experiments has several advantages, including cost-effective library preparation, starting at around $26 per sample. You need much lower read depth with quant-seq, meaning that you can reduce the read depth down to 3 million reads for standard quant-seq or 20 to 40 million reads for Slam-seq experiments. Quant-seq is a 3' protocol which has polyase selectivity, which enables you to assay functional, fully processed RNA POL2 transcripts, and there's no requirement for rRNA depletion. We also have high sensitivity and a high range of input amounts that you can use for quant-seq, from as little as 100 picograms of total RNA up to 2 micrograms. Typically, the input for Slam-seq is around 100 nanograms to 1 microgram of total RNA. Combining Slam-seq with quant-seq gives you robust quantification of T to C read conversions and covers inherently U-rich 3' UTRs. We also offer a streamlined data analysis solution, which gives you high strandedness and no length normalization requirements. The data analysis for Slam-seq experiments is also facilitated by us at Lexogen. We offer access to an automated streamlined slam dunk pipeline for Slam-seq quant-seq data analysis, which is hosted on the Bluebee Genomics platform. This pipeline is suitable for all users, regardless of your bioinformatics knowledge or experience. The pipeline workflow, as you can see depicted here, performs all the trimming, mapping, and T to C read count statistics and is specifically designed for quant-seq 3' data. We currently have pipelines available for human and mouse reference genomes, but please contact us if you would like to have other reference genomes available. You can also purchase activation codes for Slam-seq from us at Lexogen. Just contact your local sales representative or distributor. So that's all from me. If you would like any more information about Slam-seq or quant-seq and the kits that we offer, please visit our website, www.lexogen.com, or contact us anytime at these phone numbers or via email at info@lexogen.com. Thanks very much.
Thank you so much, Dr. Bannister. I'd also like to thank our speakers so much for taking the time to present today. Dr. Stefan Amiras from the Institute of Molecular Biotechnology and Dr. Johannes Zuber from the Research Institute of Molecular Pathology. Please do go to the URL that will be appearing at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This particular webinar will be made available to view again as an on-demand presentation within about 48 hours from now. And if you'd like to receive alerts about future webinars, please go to the sign-up link in the Resources tab below the slide window. We're very interested to know what you thought of today's webinar. Please send us an email at the address now up in your slide viewer, webinar@AAAS.org. Again, thank you very much to our panel and to Lexogen for their kind sponsorship of today's educational seminar. Goodbye.